Hello, welcome to my channel. I am an incoming PhD student who's spending the summer reading interesting papers, and to keep myself motivated, I make a video like this whenever I find something that I'm really interested in. It's my way of practicing talking about things that I've read and just generally, this is a very low-key channel. I literally finished reading this paper and thought to myself, oh, I feel like making a video today. So we're going to make a video. I like to present papers as if we were in a conference, in a journal club. But I learned my lesson from last time when I made a 40-minute video about a paper that I need to keep my videos shorter. So instead of presenting the entire paper today, I'm just going to present one concept from it that I find really interesting. And my goal is to keep this video maybe like 13, 15 minutes with the exception of this intro, because I'm going to tell you a little backstory about why I absolutely love this paper. Well, not absolutely love, but I really like this paper. And this is not science related. So if you're just here to listen to me talk about statistically derived geometrical landscapes capture principles of decision-making dynamics during self-weight transitions, maybe skip forward a little bit. But we're going to look at contour plots. Actually, I'm just going to show you all what we're going to be looking at. We're going to focus on contour plots like these and like this, and there's a little bit more later. Um, they are beautifully familiar to me, and this is the backstory. Because as a junior undergrad of mathematics, I took a class called Asymptotics and Perturbation Methods, which is often offered to second year PhD students at my school. And there were a bunch of first year PhD students in that class. There was me, there was another undergrad. Every single human was overwhelmed by that class. And I think the level of difficulty just exponentially increases, like the younger you are along the math tree. Um, I think it's like I think we had our semesters are 15 weeks, I'm pretty sure. I think from week 10 onwards, I just never step into the classroom without crying. That's how bad it was, and I couldn't withdraw from the class because of various issues. Otherwise, definitely would have withdrawn from the class, but I couldn't. So, like, literally the only way is through. So I tolerated that semester to the best of my abilities. And when I graduated from undergrad, I sort of took a walk around the building and I met the professor who taught the class. And he congratulated me on like finishing my degree and whatnot. And I said to him, and it is still what I genuinely believe in my heart, that that was the most overwhelming semester of my life. That was the most difficult thing I've ever done in my life. And in the future, I know that is my limit. If I ever come near to it, I will stop myself before I do so. And if I ever exceed it, I will be astonished at the amount of growth that could happen in the years between I took his class and when I decided to challenge myself again. Um, we did a lot of contour plots like this one in his class. It was my favorite part of his class. Oh, by the way, it was a difficult class, but it was an interesting class. Like I was crying because I really wish I could understand what he was saying, because the parts that I could understand was so interesting, as I'm about to share with you here. But it's just I wasn't, smart enough. I didn't have enough experience in math. I wasn't a second year PhD student. So I couldn't understand, and that's what's frustrating to me. So being able to come back, like my research is not really in math anymore, but to be able to read a paper in biology where they used a lot of the methods discussed in that class, and to be able to tell myself, hey, I understand these things, even though those were like maybe the things that I will share today maybe makes up like 5% of that grad level math class, but hey, I understand this. I know this brings me joy. I know it'll get really, really difficult later on down the road. And I know understanding this paper says nothing about how much I've grown since taking that class, because this is not a difficult paper. But it just, if the professor who taught that class is watching this video for whatever reason, that's probably not possible. But if you're watching, I have no words. Like, I feel like your class shaped who I am as a scientist, for better or for worse. A lot of the things that I learned in that class, I probably am not aware of and probably cannot put into words until much, much later in my scientific career, but it was an experience that I would not trade for anything. Like if I could go back in time and like withdraw from that class or drop the class during drop ad, I probably wouldn't do it because who I am as a person right now would just poof disappear into the air if I never took that class. Anyway, that's the emotional aspect of presenting this paper, and now you see why I am is extremely attached to this paper. Let's go into the science. Um, yeah, we're presenting this. Oh, wait, hold on. I have two disclaimers that I have to say with every video. Number one, I am not in any way associated with the writing of this paper. Like, I'm not one of these authors. And I found this paper online. I read this paper, and that's the extent of my exposure to this paper. I really hope this doesn't result in any copyright issues. Also, I am a channel with two subscribers, and whoever you are, thank you for subscribing. And my last video had 13 views. So if that results in a like copyright issue, I can't believe that that's the world that we live in right now. If you're an author of this paper and just somehow came across this video and is watching, the entire reason why I'm presenting this paper is because I admire your work. I absolutely love everything that I've read. I'm trying to become a better scientist. I'm trying to become more like you. So, thank you for producing this work. It is amazing. Mad respect to you. And I really hope you enjoy this video of my naive attempt of understanding what you have done. Second disclaimer is because I'm not involved in writing this paper, I might get things wrong. And especially for this paper, I Googled a lot of stuff and I used a lot of things I learned from that class. So my memory could be funky. I could have gotten like everything wrong. I hope that's not the case. But if what I say sounds funky to you, maybe don't trust me like an expert, because I'm not. Anyway, going back to this paper, this article is called Statistically Derived Geometrical Landscapes Capture Principles of Decision-Making Dynamics During Self-Fate Transitions. And just a brief introduction before we go into the statistical part, which is obviously what we're interested in. Self-fate transitions is well, humans, human cells, human stem cells can become different things under different stimulus, biochemical stimulus, mechanical stimulus, environmental, not environmental, all of these things can your stem cells can become a nerve cell or a muscle cell or like all of these amazing things. And that's what we call cell fate is what it can become. And cell fate transitions is just when this pluripotent cell becomes other things. It's a transition. Decision-making dynamics is the cell needs to decide what it wants to be. And that is a dynamical process. And we're interested in why the cell decided to become this. What stimulus made it make that decision? As you can imagine, that's very applicable to a lot of biomedical challenges that we're facing. So I'm very excited to be in this field. And statistically derived geometrical landscapes. Let's see where we start. Okay, so we can start with this guy named Waddington. It, I hope you can see my cursor, but this guy in apparently the year 1957 came up with this idea that self-fate decisions is kind of like you have a landscape, like you see, like when you think of landscapes in everyday life, you think of mountains and valleys. You have this two-dimensional surface embedded in three-dimensional space, which we call a manifold, and a little ball on top of that two-dimensional surface that can roll around. So if we position the ball on a mountain, it's going to want to roll down into the valley. If we position the ball in the valley, it's not going to want to go anywhere because it doesn't want to roll up a hill. Um, that's a very like illustrative way of saying this. Gravity has nothing to do with what we're doing here, but just think just think of a force field, not force field, potential field, similar to like gravitational potential that's pulling the ball downwards towards the valley and preventing the ball from rolling up the hill. That sort of potential field is what's creating this two-dimensional landscape embedded in three-dimensional space, which we call manifolds. This is I'm gonna I'm gonna break my science persona for like one more second. I used to do geometry a lot, and being forced to specify that it is a two-dimensional space embedded in three-dimensional, sorry, two-dimensional surface embedded in three-dimensional space is something that was pounded into me like indoctrination of a young girl. So so whenever I say that, I just feel like I just feel fear, like, oh no, did I do something wrong again? No. Um, Anyway, why did I even mention that? Anyway, so that's what people think cell-fate decisions are. So like it's sort of like your stem cell is a ball on a mountain and it has three different valleys surrounding it, and the ball will somehow decide to roll into one of those valleys. And at the bottom of each valley is something that the cell can be. It could be a nerve cell, it could be a muscle cell, and like all of these things. Um, so that's what we mean by geometrical landscapes. Statistically derived just means these researchers did a lot of experiments, again, mad respect, used immunofluorescence to count the proteins that existed in different stages of a cell's life, like as it approached decision making. And um, all of those statistics, along with other things that they did to mess with the cells a little bit to see what happens, becomes a cloud of data that we use some sort of statistical method. Oh, they actually mentioned it. They used the Gaussian mixture model to capture the statistical similarities between various cells. So they know, oh, all of these cells will become muscle, all of these cells will become nerves. And they have this gigantic cloud of data that they can analyze these things on. And that is the basis of what we're about to do with our landscape. So that's what statistically derived means. Now we can actually get into the landscapes. And like all videos, I tend to assume too much knowledge or too little. There's just really no in between for me. So I really, really, really hope that I'm not doing that now. Okay, I feel like the natural place to start is panel B over here. This tells you exactly how the cells are transitioning in the experiments that they have run. All cells start as an epiblast. This means they have potential to differentiate into various different things. CHIR is some sort of stimulus, like I genuinely I don't even care what it is. It's just something you can do to the cells to make it an anterior neutral or AN. It's a type of cell. Again, I don't know what that means. I'm just calling it AN. It's an end state that a cell can become. And then another thing you could do is to add in this thing called CHIR, and your cell becomes a caudal epiblast, CE, during which this process, there is a state called transitioning population, which is just their way of saying, we don't really know what this is, but our data shows that it existed. So we're just going to call it transition. I think they use TR to initialize this. And then when you become a CE, if you are continuously exposed to CHIR, you can become a mesoderm, which I think they symbolize with M, or you could become a posterior neutral, PN, if this thing called FGF is present. Um, so this is what biologically is happening. What does our data show? Oh, wait, hold on. Before we show data, I think they constructed the landscape from this biological process alone. Okay, so so they want some sort of mathematical model to describe this process. They use something called catastrophe theory, which is mathematicians long time ago just theorized like things becoming other things, similar to a network like this. And the mathematicians used a certain method to describe what they were doing, and these biologists read that method and be like, that's kind of what we're doing. So we're going to use that and see what happens. And turns out their simulations were actually pretty correct. But here, this is just a leap of faith that they did to utilize some good mathematics. And that is this beautiful panel that we have over here. Um, Oh, it's not letting me zoom in more. That's dumb, but okay. So, how do we read panel D and E? So we always read it from left to right. This is like A, B, C are different steps in time, and it is describing one singular process. So at the beginning, so for each of these graphs, the purple lines you see over here are the ridges of the mountains. So if a ball is somewhere over here, it'll want to go in this direction. because this ridge over here is preventing this ball here from going in this direction, similar to that. The black lines you see are, what they call this? Level curves. Oh yeah, they call it level curves. Basically every point along this black curve has the same height, like has the same value of a potential field. So the ball costs no energy to travel along these black lines or like within these black lines. So like if a ball were to go from this place to this place, because it's within the same black loop, it doesn't cost us energy. So the ball is able to do that. When two black lines cross, like here or here, we call that a saddle point. Um, if you remember from calculus, saddle points are when um, you have two different values of curvature that are layered on top of each other, orthogonal, and it's where let me introduce the red lines before telling you the importance of saddle points. Red lines symbolize the places and the direction with which like cells can go to. For example, if you're in an epiblast, in panel A, you can't really go anywhere because although there are red lines connected to you, both of these arrows are pointing towards you, so you can't migrate along the lines in the opposite direction. Um, However, if you're a cell on the saddle point, which doesn't have any real world meaning, but just imagine that just so that I can tell you what the red lines are, it can choose to roll down this way or that way because it is on a ridge. It is within the black circle. Yeah. Anyway, epi, AN, and CE here symbolizes three different states that our cell can be in, as shown in the graph over here. And then the idea is all of our cells started as an epiblast and at some point becomes either AN or CE. How does that happen? Well, our two-dimensional manifold in a three-dimensional space is not static. So these purple lines, these black lines, they all move um, as time goes on. They move by their movement is driven by certain parameters, and those parameters are determined experimentally. So all you are seeing here is the end result of that derivation. So we're skipping a lot of equations here, but trust me, these movements did not come out of nowhere. So basically, as time goes on, what you see in panel A slowly becomes panel B, and panel B slowly becomes panel C. Now, the major difference between panel A and B, as you see here, is that the epiblast is at the same level as AN and CE. Like there's no little black blockage over here that's preventing the red lines from like um, being on the same surface. So panel B is when epicells can decide whether they want to become AN or CE. And wait, hold on. Did I get this right? I think I got this right. And then as time goes on, this little border happens again. So in panel A, your epi cell can become AN for free. It cannot become CE. In panel B, both can happen. In panel C, your epi cell can become CE and it cannot become AN. And whenever a cell becomes AN, it sort of stays there until panel C when it can no longer return to epi. And similar to CE, at this point, it is not possible to become it, and now it is possible. Does that make sense? Like, I hope that made sense. They call this binary choice because you are either AN or CE. Because as you see here, we're creating AN, you cannot create CE. We're creating CE, you cannot be AN. That's what we call binary choice. Your epi becomes one or the other. And this is just the transition state when that happened. This is instantaneous. So this is like a split second when both can become both. And then after that point, it continues to be binary choice. Panel E describes the second half of this graph. So that was this part of the graph. Now we're describing this part of the graph where we have three nodes. And I feel like it could be a good exercise to like, I don't know, read this and see whether you understand it. So pause the video if you want to do that. Welcome back. Um, so starting with panel A, we have a bunch of CE cells, which is what we left off on this panel over here. All of our cells should probably be like over here. And here they can choose to travel this way. No, it cannot because this was blocked. Can they choose to travel this way? No, it cannot because there's not a red line over there. And then let's skip to C for a bit. So when you compare panel C and panel A, you notice that the purple lines have flipped, like quite literally. And that's why we call it a binary flip, because our potential field literally flipped. On our panel C, can our panel CE become PN? No. But can PN become M? Yes, because this is the same playing field and it passes through a saddle point. And now we come to the enigmatic panel B. Panel B is the only instant in this entire timeline when the CE cells can do something because it is on the same playing field as everything, basically. And this line over here is red and purple. Basically, I think this line here just slowly started approaching the middle of this graph until at this point it is exactly aligned with the saddle point over here, and then it ticks upward and becomes this line over here. And when it is exactly here, imagine like this is the ridge of the mountain as we mentioned. So CE is finally on the same playing field as this saddle point over here. So CE can travel from here to here for free because of the properties of the mountain ridge. And from here, it can decide to become M or PN because both of these are within its region of reach. So at this point, because of statistics, it becomes a 50-50. So 50% of CE cells become M, 50% of them becomes PN. And as time goes on, we come here, PN and M can still communicate, but if there are CE cells that were left behind in this process that didn't come over here, they are stuck here forever. In reality, that doesn't happen much, but I think it does. I don't know. It's like statistical noise at this point. But this graph describes everything that is happening here, especially this double arrow over here where they can still become each other after this initial signal. Um, I don't think there's, oh, what, right. And we call this a binary flip because in the end, we end up with 50-50, excluding communication among them later on. So binary choice is when you have all or nothing, all AN or all CE, and binary flip is at like one instant, suddenly it's 50-50. So that's two different modes of operations that like logically should exist. Because if you logically think about like one thing becoming another, you think there are two ways that that could happen. One is like everything that I have becomes that other thing, or 50% of what I have become that thing and 50% becomes another thing. That's just logically two things that could happen. And math here is just to describe all of that in a graphical way so that it's very directly understandable. And also it introduces a potential field, which is interesting to us mathematically later. Um, I think that's everything that I want to mention with these graphs. And then panel F over here is just the combination of panel D and panel E connected by this CE region over here. Everything that I said about time variables here will apply to this graph. This is just a snapshot of what the system looks like at a specific moment in time. This seems to be, I think, the starting point. No, actually, this should be towards the end. You know what? I'm not really sure, but this is basically the conglomeration of everything here. What does X and Y axis mean in this graph? Essentially, nothing. X axis is roughly the time variable because like most times like we like decision making happens from left to right in our graphs, and Y axis is roughly like just a sorting mechanism. Generically, this entire graph has no physical meaning. It just tells you the statistical distribution of where cells want to go. And each point on this graph, well, most points on this graph has no meaning. but the points that have color and are solid here represents like the states that the cell could be in, like whether that's CE or M or PN, is just something that could happen. And I think that's all I wanted to do with those graphs. Okay, and then they started simulating with math. So they put a bunch of cells in epi-state over here. They dragged the time variable along this axis, and they documented how many cells wants to make a decision to become this or that as time goes on. And as you see, the black contours here changes shape as time goes on because, again, our two-dimensional manifold is changing shape. And that's sort of the entire reason why self-fate decisions are happening right now. So what's driving this dynamical system is time, which is correlated to manifold shape. And then they ran that simulation and they compared their simulation to experiment in terms of like how many cells that are in which state. And most of the time they fit pretty well. So this is their step to verify that yes, this brainchild of a mathematician from hundreds of years ago in terms of logic and simplicity actually applies to this self-fate decision thing that we are trying to tangibly understand because their predictions align with experimental results. And I think that's it. There's nothing else here that I want to mention. Pretty graph, good graph. Yeah. Well, thank you for observing this channel. Um, I hope I see you again in the future. And if I don't, um, that's okay because this channel is just for vibes. All right.